We all strive to live a long, prosperous, and healthy life. With advances in health and medical sciences, this goal is ever more attainable. The Sam and Rose Stein Institute for Research on Aging is a non-profit organized research unit under the auspices of the University of California at San Diego, committed to advancing lifelong health and independence through research, education, and patient care. To better empower and improve the lives of young and old alike, the Stein Institute presents the following program.
So tonight's speaker is Dr. Nazneen Dewji. She is an associate professor of medicine here at UCSD. She was born in Tanzania and raised in England. She attended the University of London, where she earned her Bachelor's of Science in Biochemistry and her PhD in Protein Chemistry and Enzymology. She joined UCSD in 1982, where she did a postdoctoral fellowship in the Department of Neurosciences, working on the molecular genetics of inborn errors of metabolism. In 1986, Dr. Dewji accepted a faculty position in the Department of Medicine here at UCSD and has worked on the molecular and cellular mechanisms of Alzheimer's disease since that time. Tonight, she's going to be presenting her research on the novel approaches to the treatment of Alzheimer's disease. So please join me in welcoming Dr. Dewji.
Thank you, Jody. It's a pleasure to be here, more so because this lecture series was started by J. Siegmiller, for those of you who remember him. And J. Siegmiller was the man that hired me as a junior faculty in the Department of Medicine. And in fact, I started work on Alzheimer's disease when I was still in his lab. He had provided me with space in his lab. So it's a particular pleasure to be here. All right. Let's see if I can get this going. Just a disclosure that I'm one of the founders of Sena Biosciences, which is a local company. So Alzheimer's disease is a progressive neurological disorder that affects people in all parts of the world. And age is a risk factor. It's the major risk factor for Alzheimer's disease. This slide was first published by Bob Katzman. I think I should take a moment to remember him. Bob Katzman passed away yesterday. And he is one of the people that made a major contribution to Alzheimer's disease. He and Bob Terry actually defined Alzheimer's disease. So I thought it was appropriate to say that. Okay, so age is a risk factor. 1 in 10 people over the age of 65 get Alzheimer's disease. But the incidence increases logarithmically with age, exponentially, so that at age 90, there's a 1 in 2 chance of getting the disease. So that becomes a huge problem because the largest segment, the fastest growing segment of the population is 85 years and up. And by the year 2050, roughly a quarter of our population should be age 85 and up. So this is an enormous problem. And of course, it's a problem because of the success that we have, the success we've had in providing good health care and nutrition. The morphological changes in the brain in Alzheimer's disease include cortical atrophy or degeneration of tissue, brain tissue, loss of neurons of specific cell groups, and dysfunction and loss of synapses, and also the presence of neurofibrillary tangles and neuritic plaques. And I'm going to go through each one of those and explain what they are. So this slide shows a normal brain, a cross-section of a normal brain, and an Alzheimer's brain. And these areas in red show the tissue degeneration in Alzheimer's disease that's not present in the normal, okay, or very little in the normal. And brain changes don't take place all over the brain. They appear first to involve an area of the brain called the entorhinal cortex, and then the hippocampus. The entorhinal cortex is the area in yellow, and the hippocampus, which is the area in red, and later the frontal cortex, which is these are the areas of the brain that are involved in memory, and that's the reason why the memory goes first in Alzheimer's disease. I said there was a loss of synapse as well. Synapses are areas of communication between two cells. So this would be one cell, and that's another, and that's a synapse. That's a synapse. And because of shrinkage and degeneration, there's a loss of synapse in Alzheimer's disease as well, and mostly in the presynaptic terminal. I said that there was the presence of amyloid plaques and neurofibrillary tangles, and this is what a normal brain would look like. These are the neurons, and there's nothing else, okay? In an Alzheimer's brain, you'd have some neurons that are filled with neurofibrillary tangles, and then there is the extracellular deposit of plaques, of which the central core is made of amyloid. And I'll show you some more pictures. So this slide is a slide of a tangle that's been stained with a fluorescent stain. And you can see that tangles usually take the shape of the neuron. In this case, much of the neuron is filled by these wispy structures, these fibrous structures. And tangles were known even at the time of Alzheimer himself. This is a picture drawn by Bonfiglio in 1908 with a silver stain. So these are tangles. Okay, this is a section of the brain, of a region of the brain, the amygdala, from a 67-year-old man with a progressive history of dementia. This is stained with silver, and what you see here. These are neurons that are filled with tangles, these dark structures, whereas a normal neuron would be this golden structure. And in the center is a large plaque. These black structures are degenerating nerve terminals, and in the center is a large compacted disc of amyloid. So if you stain plaques with Congo red, which is another fluorescent dye, and you look under a certain type of microscope, a polarizing microscope, then you see this classic birefringent Maltese cross. These are plaques. This is amyloid in the plaques, in the center of plaques, and they appear like that. And amyloid is also found coating the walls of the blood vessels in the brain. Okay? And you know, amyloid was found right here at UCSD. It was discovered here by George Glenner, who some of you might remember. And really, George Glenner made an extremely important contribution to Alzheimer's disease because the majority of work that's being carried out right now is on amyloid. And it's important because beta amyloid, and I'll be talking about beta amyloid, which is the same thing as A beta, so I might use both terms and they mean the same thing. Beta amyloid is considered to be the major toxic species in Alzheimer's disease. This has come after years of work, okay? There are two major forms of beta amyloid, or A beta, comprising of 40 and 42 amino acids. And the 42 amino acid, the longer A beta, is considered to be a little more toxic. The amyloids are both 40 and 42 are very insoluble, and they aggregate. They're very sticky as well. And I've just shown you that amyloid accumulates in the brain in plaques, and in cerebral vessels. So I told you that beta amyloid is considered to be the toxic species in Alzheimer's disease. And it starts out, and some work in our own laboratory has shown that it starts out by making holes in membranes. That's one of the ways A beta is toxic to the cell. But after it gets deposited, and you can see that deposition starts much earlier than the symptoms of dementia. Okay, so once it gets deposited, after a while, after several years, you get microglial activation. So microglia are scavenger cells, and they're usually found surrounding amyloid. And so that's the reaction, the natural reaction that the body has, the brain has, to activate these cells. And then you see the presence of neurofibrillary tangles, which I've described. Then comes the neuronal loss and neurochemical changes that take place in the brain. And finally, you get dementia. Now, we have a problem in that there is currently no cure for the disease, as you all know. There are only two classes of FDA-approved drugs, and they are acetylcholinesterase inhibitors, and another one that's an NMDA receptor antagonist, Memantine. The best thing they do is they provide symptomatic relief for a very short time. They do not correct the basic pathology of the disease. So you can see here, disease-modifying therapies, there's nothing at all, whereas there is some progress being made in taking care of symptoms for a little while. And these cholinesterase inhibitors, such as Aricept, for instance, they provide just a slight improvement for about 6 months. But after that, they just stabilize. And so this line is a patient with Alzheimer's disease who doesn't take any treatment, and they go down. And this is somebody who's on something like Aricept. And you can see that you still go down. So it does not, what we need is disease-modifying therapies. So the clinical goals for disease modification are to extend the period of healthy older age, delay or prevent the development of symptoms, and delay the use of symptomatic treatments such as Aricept. So, for instance, the goal is that if you, in this case, this blue line is an untreated patient. Right now, we have drugs that give symptomatic relief, but you can see that still there's a decline. And this is what we're aiming to get at, to develop disease-modifying therapies which will really change the course. They will stop the decline. Okay. So how does one do that? Well, one of the things that you really need to do is to understand mechanisms that give rise to the toxicity. And I thought it would help to show the slide of a timeline, because that'll help us understand how we, at least our approach, to determining the mechanism that produces beta amyloid, because that was where we started out from. We wanted to learn how A beta was produced, the mechanism that produced A beta, so that we could intervene. All right, I thought let me just start with this, gentlemen. I thought it would be interesting to let you know that Avicenna or Ibn Sina actually described dementia 1,000 years before Alzheimer did. And Alzheimer described his first patient in 1906. The three most important discoveries that have happened that have led us to determine what the mechanism of A beta production is. Well, first of all, in 1984, the discovery of beta amyloid by George Glenner. Then in 1991, the discovery of mutations in the beta amyloid precursor protein gene in families. So the discovery that if you had a mutation in the beta amyloid precursor protein, which is the precursor to the A beta, and I'll show you a slide later on, then those families got Alzheimer's disease. And the third discovery was a discovery of two other proteins called the presenilins, PS1 and PS2. And these presenilins were discovered in families where a mutation in these genes gave rise to the disease. So again, we're talking about familial Alzheimer's disease, so the genes that are responsible for familial Alzheimer's disease are beta IPP on chromosome 21, which comprises of about 2 to 3% of all familial cases, very small number, and only 1% of all Alzheimer's cases. And presenilin 1 and 2, which presenilin 1 is really the majority of all familial Alzheimer's disease cases, and presenilin 2. And these were discovered because people were looking at families with Alzheimer's disease. Okay? So, and you can see that one of the things is quite clear is the age of onset is much earlier in these familial cases. So even though the familial Alzheimer's disease cases make up really a very small percentage of all Alzheimer's cases, they give us a handle. They give us an opportunity to study the mechanisms that would be involved in all Alzheimer's disease cases. Okay, and that's what we've done. So let's start with the first one, the beta amyloid precursor protein. The beta amyloid precursor protein is really the parent molecule from which A beta is cleaved. So A beta here is the red box. And A beta has to be cleaved by enzymes called the secretases. So this would be a beta secretase over here and a gamma secretase over there to produce this red box, beta amyloid or A beta, which is the toxic species. And beta amyloid precursor protein is a special kind of protein. It's a membrane protein. Not all proteins are in the membrane, but there's a special group of proteins that arise in membranes, and beta amyloid precursor protein is a membrane protein because it crosses the membrane one time, as you can see. Now, the presenilins are also membrane proteins, but they go through the membrane multiple times. So that's the structure. That's the structure of presenilin that we think, or we have evidence for, and that is a protein that goes through the membrane 7 times. Now, other groups have shown that this protein goes through the membrane 8 times, and some more people have shown that it goes through 9 times, and still others 6 times. But our evidence suggests that this protein is a protein that goes through the membrane 7 times. Okay, and I'm saying this because it'll become relevant as we go further. What is the relationship of these proteins? So when the presenilins first were discovered in 1995, John Singer, who's here, and I'll show you his slide. John Singer and I were working together, and we asked, how is A beta produced from beta IPP physiologically? And how do presenilins, PS1 and PS2, participate in this process? Because you see, if you have a mutation in the beta IPP gene, or you have a mutation in the presenilin gene, you still get the same pathology. You get the deposition of A beta. So how are these proteins related? And that was the question we asked. And before I go into it, I want to just say that most people today, currently, most investigators believe that these proteins interact with each other, but that interaction takes place in one cell, on the same cell. And our proposal was different. It was different in that this one has green proteins at the surface, and they are PS1 and PS2. And then there is another cell, and it has red proteins on the surface, and that is the beta amyloid precursor protein. And what we suggested was that there was an interaction between the presenilins and the beta APP, but this interaction utilized two different cells. So it was a cell-cell interaction, rather than everything happening in one cell. And we suggested that, so this would be presenilin, this is one cell surface, and that's another cell surface expressing beta APP. And we suggested that these proteins would interact with each other in a cell-cell interaction, and that this cell-cell interaction was necessary for certain normal responses, certain normal physiological responses. But that as a byproduct of this cell-cell interaction, the proteins still bound to each other would be taken inside the cell, and then A beta would be broken down inside the cell. The beta IPP would be broken down to A beta inside the cell, okay? So the main take-home message is that we suggested that there were two cells involved rather than one cell. Okay, so we spend the next 10 years or so trying to prove that. And I'm only going to show you one set of data, our latest one, which shows that indeed the cell-cell interaction is necessary for the cell-cell interaction between the presenilin and the beta APP is necessary for the production of A beta, which is the toxic species in Alzheimer's disease. Okay, so what we did was we engineered cells so that they would only express one protein. And this cell is a mouse cell that expresses the human PS1 or PS2. And we engineered cells so that they would only express mouse cells expressing human beta APP. But there's no presenilin here, and there's no beta APP here. And what we did was we mixed them. We cultured them together. And then we looked for the production of A beta. And what we found was that only in those cultures which had the beta APP expressing cell and the presenilin expressing cell did you get the production of A beta. But if you replaced one or the other of these cells with a cell that didn't express that protein, then you didn't get any A beta produced. So that suggests that you require both the presenilin and the beta IPP to produce A beta, and that these cells undergo a cell-cell interaction to do so. Okay? All right, so we did a couple more experiments where we took these beta IPP expressing cells and the presenilin expressing cells, and we cultured them at high densities so that all these cells are now touching each other so that they're going through a cell-cell interaction, right? Or at lower densities where there is less and less contact. And then we made cell extracts, and we took exactly the same amount of protein from each one of these. So the only thing that was different in our samples was the way the cells were plated, whether they were touching each other or not. And we looked for the production of A beta. And we found that the A beta production was highest in cells that were the densest, and that it went down progressively. And in this case, where the cells are hardly touching each other, there is no A beta produced. Okay. Now, in nature, though, you remember I told you we engineered cells so that they expressed only one or the other protein. But in nature, both these proteins are present on the surface of cells together. So we asked the question whether a similar cell-cell interaction was taking place even in cells that expressed both proteins. And so we prepared cells so that they would express both, this time both the beta-IBP and the presenilin on the surface of the same cell, and then we cultured them so that they were touching each other at high density or at lower densities where they were far apart. And in fact, the result we got was identical to the one where the cells were separately expressing each protein. So that you now have, exactly what is happening is that even in cells that express both the proteins, a cell-cell interaction is taking place to produce A beta. Why is it so important? I mean, who cares whether it's the same cell or two cells? Well, the important thing to remember is that if you have two cells that are involved in the production of A beta, then if you found a way of inhibiting that cell-cell interaction, then you might find a way to inhibit the production of A beta. And that's exactly what we did. And in this case, we inhibited the cell-cell interaction with the region of the presenilin that is extracellular. So we made a lot of it. This is the amino term. This is presenilin, goes through the membrane 7 times. And this is the extracellular. So this is outside the cell. This would be inside the cell. And what we did was we made masses of that. And then we added that in the cultures so it would act as an inhibitor, because that would preferentially bind the beta-IBP. So the beta-IBP would no longer be able to bind the presenilin. Okay, so it acted as a competitive inhibitor. And what we found was that if you throw in, and this is again another co-culture, the cell-cell interaction that have shown you before, if you carried out a cell-cell interaction between beta-IBP and the presenilin, you get A beta produced. But if you throw in this inhibitor, then you inhibit specifically the production of A beta. And if you make a non-specific inhibitor from another presenilin, like PS2, then it doesn't inhibit A beta production, suggesting that this inhibition is very, very special, very specific. Okay, so if you can do that, then you can start thinking of making drugs. So the summary is that both A beta, both the species, the 1 to 40 and the 1 to 42, can be produced from the cell-cell interaction between beta-IBP and either PS1 or PS2 in cells that are plated at high density, and that you can inhibit this production if you inhibit the cell-cell interaction between the PS1 and the beta-IBP. And cells that are co-transfected, cells that are expressing both the beta-IBP and the PS in the same cell, when plated at high densities, also produce A beta by the same kind of cell-cell interaction. Now, what's interesting is what I have shown you so far is work that's done in tissue culture. Okay, that's not in an animal. But we now have some preliminary data in animals, in transgenic animals. And these are animals that produce the Alzheimer's, these are animal models of Alzheimer's disease. They produce the same A beta. And if you give them this inhibitor, then you get a substantial reduction of the A beta. So we are now engaged in trying to develop this for a drug. Okay. I would like to talk to you now about another target. So this was one target. And I talk to you about the structure of the presenilin, because that comes into it. Our evidence suggests that the presenilins go through the membrane 7 times. Why is that important? I mean, some people say 8, some people say 9. Why is it so important? Why is a 7 transmembrane protein important? Well, there is a family in nature, it's the largest family of proteins called the G protein coupled receptors. These are proteins that are very important for many functions in the cell. They all of them have 7 transmembrane structures, so they all go through the membrane 7 times. Now, not all 7 transmembrane proteins are GPCRs, but all GPCRs have a 7 transmembrane structure. Okay? And what's important about them is that GPCRs make very successful drug targets. We know how to develop drugs from that kind of structure. And greater than 50% of all drugs that are made today target GPCRs. So if the presenilins can be, when we started this work, we didn't know whether the presenilins also couple G proteins. That is, even though we had evidence that they had 7 transmembrane structure, we didn't know whether they were also GPCR. So we did some work, and in fact, what we found is that both PS1 and PS2 do couple G proteins, and that's G protein GO. And we have work that's ongoing on the G protein G protein coupled receptor function of the presenilins. But one thing that we're working on right now is that if you now interfere with the GPCR function of the presenilin, which we know how to do, then you could possibly inhibit the production of A beta in that way. So that's another way of doing it. It's another target. And our approach now is to inhibit the production of A beta by specifically inhibiting the cell-cell interaction between beta-APP and PS, as I've shown you before. That's target 1. And to inhibit A beta production by modifying or inhibiting the GPCR function. This is the 7 transmembrane story of PS, of presenilin. So, but what we think is that a combination of the 2 approaches may actually be a little better. So we're following both the paths right now. And the advantage of this approach